It's a great pleasure to be here with you today, Dr. Paul MacLean, to talk to you about your contributions to neuroscience, your life and your work in understanding the brain and mind, and how you have developed the concept of the limbic system, particularly with regard to the role of emotions in the functions of the brain. And I think a good place to begin is from the beginning. How did you start all this? How did you begin to get interested in this work?
That's a hard question to answer, but it goes way back. But I will start later, and it was just after I graduated from Yale in 35, and I had my ticket all bought to go to Edinburgh to study philosophy under A.E. Taylor, the great Plato scholar. And shortly before I left, my mother was taken acutely ill, and this wonderful surgeon in Genesee County, Batavia, New York, where we were living then, came to the house, and we thought she had a heart attack, and he said no, she had a gallbladder attack and was going to get over this very quickly. Well, that was very reassuring. And I thought to myself, my God, why am I going to study philosophy and read philosophy and just rehash what philosophers have been saying for 1,000 or more years? Why not go into medicine and find out what is really up there ticking? And how did we become, the thing that's always, I've always wondered at is how human beings, as such rational individuals, can be so irrational at the same time. And I'd have to go back and tell you a story about when I was this high and sitting alone in church and having my father call out my name to clarify that. But there I was, my mother and two older brothers sitting in the choir, and a little piece of plaster fell in the church, and I was sitting in the back, and I was going over the partition in the pew, and I heard a name call out, Paul. And that was my father telling me, what are you doing? So right after church, I got on my tricycle, and I was just going to run away from home. And they sent out a searching party for me, and my two older brothers found me down at Anna Kent, our dressmaker, who was very understanding. So that's basically how I got into medicine, was through my mother's illness. It just stirred up these original thoughts of how do we have these compulsive feelings to do what we do in spite of our rational nature. And you were going to ask me another question, but you know, the people who subscribe to Spinoza, that men are governed, I would say human beings are governed by their emotions. I thought this was the probable explanation, but we've gone a little deeper since then, and I would say the reptile is a little more important in this business.
And you have basically spent your life in trying to find the source of these actions which at one level to an external observer look like they're very impulsive, but they're not emotional in this common sense of the word.
Yeah.
They are basically almost a reflex, a very mechanical kind of response, but uncontrollable once they start, and others are not. So this interaction between the various parts of the brain is, of course, is what has led you to this, the theory of the reptilian brain.
Well, that's how I got into emotions, first of all, and for many years, as you know. But the reptile, it seems, does what it has to do, and that the emotions, more often than not, it seems to me, are merely reflectors rather than determinants of action. That's how I would differ a little from the Spinozan view.
This leads me to the next phase in your life, because you basically then had to join the army for a while. Your life was changed when the war broke out, and I think one of the formative influences was your experiences in a psychiatric hospital. Perhaps you can tell us a little bit about that and how it led to the next stage in your life.
Well, actually, I went to Yale Medical School, and there with the reflexology that was being taught at that time, even with John Fulton, who was there as a professor of physiology, and who really sort of put his finger on the frontal lobes. They were still talking reflexology, and I decided that probably the mind wasn't in the head after all. And so I prepared for a practice in cardiovascular disease. And I had gone to the Hopkins and was on my way to Boston next, Peter Bent Brigham, with, where Soma Weiss was teaching. Well, he died, as you know, of a subarachnoid hemorrhage, and war was declared. And so I went with the 39th General Hospital to New Zealand, and we thought we were going to Alaska, but we ended up in New Zealand. And there I was doing bacteriology, and Averil Liebel and I had identified this diphtheria bacillus in these cutaneous ulcers, which the soldiers were suffering from at that time. Up in the tropics, the skin was very much like the mucous membranes of the nasopharynx. Right. These poor fellows, they would get these stocking anesthesias too and would be sent home as hysterics. But I wasn't in the lab long when the psychiatrists all moved up to the front office, and Merrill Moore was the only psychiatrist left, and they needed someone to take care of the disturbed ward. And since you didn't have to know anything to take care of disturbed patients, I was chosen, and was pretty much of a novice, I could assure you. And but I learned very rapidly, especially when we had one soldier had to remove all his clothes and put him in a padded room. And I went in there one day and told the crew to stay outside, the nurses and the corpsmen. And I was standing in there thinking that I was going to help this fellow by just showing my trust and all. And I suddenly felt I couldn't get any wind, and he was pulling both ends of my tie and choking me. So fortunately, one of the corpsmen saw me through the little window and rescued me. Otherwise, I might have been like Gudin, the Mad King, who he drowned in rescuing him, you know.
This phase really then ended when you came back. Now you're obviously very interested now in psychiatry, in the mind more directly, and apparently this was when you started to plan to get into this more closely and went to Boston.
Well, not right away. When I got out of the Army in '46, I was I had this good friend, Jim Haviland, who was living in Seattle, Washington. He said, "Paul, if you want a good life, you ought to think of Seattle." And so as soon as we got out of the San Antonio hospital, we went west and looked at San Francisco, and then we thought we ought to look at Oregon, and then we thought we ought to look at Seattle, which we did. And we loved Seattle, and we decided to settle there and get into practice of medicine there. And I did there what nobody else did, and that was set up practice in our own house. And we fixed it up, and there weren't any nails or anything those days, so we just had to glue this place together. And but you know, I was brought up to do a careful physical examination at Hopkins and take a very careful history course. And I was charging the unbelievable prices of $3 an hour, which undercut my colleagues in town. And still, I wasn't finding anything wrong with most of my patients. And I'd end up by giving them elixir of phenobarbital or chloral hydrate to help them sleep. And this didn't seem to me a satisfactory outcome. I'd learned just enough of psychiatry in the war to make me think I better get some more background in psychosomatic medicine. And I had admired Stanley Cobb's writings, and I wrote him and asked if they might have a position for me. And at the same time I had written the letter, I went to the Atlantic City where the medical meetings were in those days. The American Medical Association met there. And damned if they couldn't find me, they couldn't locate me, and they were telegramming me to come up for an interview, and I wasn't getting the message. So I finally did get to Boston, and I was offered this public health fellowship, which was just wonderful.
So now we come to your first period in Boston, and in which you basically were now on a U.S. Public Health Fellowship in the Department of Psychiatry.
Right.
And this was a very critical time because you read the famous paper by Papes, you even met Papes, and you developed your nasopharyngeal electrodes, which I think were a very basic tool in your first explorations of the brain. Can you tell us about that period?
Well, I hadn't met Papes when I went by the time I went to Boston, but I met him afterwards. And as you know, and as everyone knows, the hypothalamus in those days was considered to be the center of emotions, right? So I thought, well, why not record from the hypothalamus? And Grinker in Chicago had done a little recording with a nasopharyngeal electrode. But all the electrodes that we had at that time were quite noisy, and I devised a very simple nasopharyngeal electrode and put it up my own nose, so I knew that it wasn't that uncomfortable, and we could record from patients. Well, I discovered belatedly that I wasn't anywhere near the hypothalamus, 6 centimeters or something like that away. But I was very close to the temporal lobe, and we had had a lot of patients in the Schwab laboratory because they were interested in the EEG and these temporal spikes. And I discovered that the nasopharyngeal electrode recorded these great explosions in the medial temporal lobes. But more puzzling to me was that these patients complained of every kind of sensation, every kind of aura affecting visual, auditory, somatic systems. And I was recording from structures which were supposed to be part of the rhinencephalon. And I thought, how in the world can these patients have all these symptoms with these explosions going on in the rhinencephalon? I first met Papes at this poster presentation that I had at Atlantic City in June of '48, and I had already knew of him because I had read his paper, and it had been about his proposed mechanism of emotion, and it explained how the rhinencephalon, instead of just being a rhinencephalon, was involved in emotion because it had all these connections with the hypothalamus. And the cingulate cortex was a receptive area for emotion in very much the same way that the visual cortex was a receptor for visual information, you see? Well, this was fascinating, but except for a few suggestions, it wasn't at all clear how these different sensory systems were playing into the limbic lobe. And I asked Cobb about this later, and he said, well, why don't you go up to Ithaca and call on Papes and ask him? And this is what we did. This is what I did, and my friend and colleague, Indian from South America, we made this trip in November 1948.
And that's really was one of the starting points for your work, which went on in Yale in the John Fulton labs, and you then started to ascend the academic ladder and became associate professor of psychology. Physiology, sorry. And had plans of really remaining there, working on this problem. But then you began to get calls from elsewhere, particularly from Zurich and also from NIH.
Well, let me correct that by saying I wasn't called to Zurich. I wanted to go to either Zurich or to Freiburg, Germany, and I applied for, through the support of Dr. Fulton, I applied for a fellowship from the National Science Foundation, and it was a very generous senior postdoctoral fellowship. Just let me retract a little bit there. When I was up at Cornell, and Pepys was just a wonderfully giving person, and saintly in many ways, I would say. And with these two novices, he would just gave up all his time for 3 days to walk us through the brain. And it was just like a walk through a park. He knew it so well, and we had these macaque slides and so forth. And we just went through the whole brain that way. And I said I felt I learned more anatomy during those 3 days than I did have ever since, but that's not quite true. But he was puzzled about these. He talked of the hippocampus as though it were a little factory manufacturing emotions that were out of thin air, and then this was passed on through what has been called the Pepys circuit up to the Cingulate Gyrus, which took in all this information. But while I was there, he was pointing out how visual and auditory and somatic stuff was getting into the hippocampus. And really, this was the only thing I added later when I wrote the visceral brain paper was these connections that he had talked about. But he would, his family were Papists, and they were, I think, a little persecuted in their time. And whenever he got onto a subject that was quite not orthodox, he'd always come up close like this and begin to whisper, "What are they doing there?" he said.
Yes.
"What are they doing?" And so when I just assumed that they were coming in through cortical association pathways. But actually they were, as we followed this up and discovered later, they were coming across cortical systems, but also coming up from below with brainstem inputs from the visual, auditory, and somatic systems.
And then you, Dr. Wade Marshall at the NIH had been contacting you about the possibility of moving to the newly established laboratories at the National Institutes of Health in Bethesda, and you finally succumbed and moved here to this area. Tell me about that and the first work in that exciting lab that was really a joint effort of the Mental Health Institute and the Brain Institute at NIH.
Yeah, I'm not sure this didn't begin in the early 50s when Jack Mazur came up to New Haven. He was looking for people to fill these beautiful big laboratories they had down here. And I was just out of the Army, and I wasn't about ready to get connected with anything governmental again right away. But, you know, this was the beginning of the golden age, really. I mean, the 50s have to be called the golden age, and as far as the National Institutes of Health are concerned, certainly. And we were having this wonderful support. But it wasn't until I went to Zurich in '56 and spent a year there that I got separated. I sort of saw New Haven as my final resting place. And I think the trip to Zurich made it possible to make that break. And you had people leaving the universities to take government jobs. A number of my friends had come down to the NIH at that time, and I'd gotten to know Seymour Ketty, and then Bob Livingston, and Wade Marshall was, you know, he was wonderful. We were talking earlier about the freedom that we've enjoyed, and as I said, there have only been 2 times in my life when somebody's told me what I should be doing in research, and that's just marvelous, isn't it?
It is.
Be pretty hard in this managerial system we live in
Almost
today.
impossible to do, and to start something new.
Yeah. God.
This was in 1957 that you began working, and actually since then, from 57 to now, you have remained at Bethesda through a number of different positions, but your work has remained consistently in this area. And I think we're coming now to really the crux of the matter, how you moved from a general interest in the subject of emotions and behavior and the neuroanatomy of the brain to really linking this to evolution of the mind and the brain together, and to your theories, particularly the theory of the triune brain. And perhaps we can have that story unfold now, because you're now in one place and you've worked consistently for over 30 years in this field.
A great stumbling block for me was a very large piece of soft tissue in the center of the brain, which is the caudate nucleus and company, the corpus striatum. And it was so puzzling. Back in the MGH days, I was mentioned Charlie Kubik. He would come in from Lincoln and I was coming from Concord. We'd meet at the North Station, then walk up those back alleys to the MGH at that time. He was a neuropathologist. He'd seen all these patients come to postmortem and had these large, they'd have large holes in the caudate or putamen, but no motor symptoms. And of course, the traditional clinical view, and it still is pretty much, is that the corpus striatum and company is the highest level of the motor apparatus, under the control of the motor cortex with no mind of its own, so to speak. And we'd get these, I'd been to Scotland, as I mentioned, and I was very well acquainted with the smell of meat as it was hung out to cure. And we'd go by these butcher shops and smell the meat, and this somehow had a way of kicking off this question about what are the functions of the basal ganglia. And I said to Charlie, as I since called him, what do the basal ganglia do? And he said, oh, they don't do nothing. They just hold up the rest of the brain. Well, I was rather reluctant to go along with that until I went to Yale, and when we were doing chemical and electrical stimulation of the brain, using that method, which we'd devised there, I found I was sometimes right in the middle of the caudate and kicking it as hard as I could with either electrical or chemical stimulation, and not much was happening. So this became a real puzzle. And if I'd stayed at Yale, I would have, I'd hoped to have a field laboratory where I could look at animals in their natural environment, doing what they want to do, rather than bringing them into the classroom and putting them through reading, writing, and arithmetic, the usual thing, you know. And so it happened that Bob Livingston, who was director of the both institutes, as we were talking, mental health and neurology, he was very sympathetic to this idea. And there was Calhoun, who was out already in the Casey farm doing the work with rats, so it seemed very reasonable to him that when the NIH was planning an animal center, well, for heaven's sakes, let's get a piece of this for mental health and have this field laboratory there. And this started, and we began talking about this in '59, and the whole thing dragged on, as you know, for over 10 years. And That was because we were a little small part of the package, and they kept changing the design and so forth for the NIH Animal Farm. But we finally did get this laboratory in '71, the laboratory that you are sitting in right here. On this lower shelf are animals in which I'd produced lesions and had shown that the globus pallidus in the squirrel monkey is quite essential as a site of convergence for mechanisms involved in the natural display behavior of the squirrel monkey, and which has both autonomic and somatic components, which makes it very much interesting. And also from the cognitive point of view too, you've got to recognize another monkey if you're going to display to it, right?
Right.
But I was still, in a comparative sense, I wasn't studying the corpus striatum, and I thought it was only meet and proper that we get look at reptiles, you know. And finally, in '75, we brought a man here, Neil Greenberg from Rutgers, who had done his thesis on the blue spiny lizard. And we got into actually observing reptiles and what they were doing. So we looked at reptiles and lizards, and I chose lizards because they seem to come closest to resembling the mammal-like reptiles, the therapsids. We had the blue spiny lizard, but we didn't know much about all these other different kinds of lizards, so we looked at quite a variety and decided that the little common green anolis lizard would be a very good subject. But then we had to go on and devise microsurgical methods and all to make lesions in the brain and so forth. Well, the sum and substance of it all is that we spent an awful lot of time looking at lizards, and I began to become aware of things that I hadn't been aware of before, and that is how important it is to have a brain that can put together the daily routine, daily master routine, and all these little subroutines. That if a female appears, then you have to break the daily master routine to court the female, right? And so this was a great eye-opener to me, and I was fascinated later to discover that in Huntington's chorea, that one of the first symptoms the patients have is they can't put their daily routine together, but they're delighted to have somebody to follow and carry out a routine that they can mimic. And the same with, as far as subroutines, the Sydenham's chorea, which is presumed to affect the corpus striatum. The evidence isn't all that great, but you know, they have these subroutines. The Osler describes these cases. The one girl had to knock up the door 100 times before she could enter, and then she had to brush her teeth 100 times.
Compulsive, obsessive, compulsive behaviors.
Yeah, and these are very disrupting, and particularly not only to the patient but also to the people who are caring for them. And of course, the psychotic patients, they get into these subroutines, which are just consume all the family's energies and the nurses' energies, because they aren't sick in bed like someone on intensive care. They're walking around free, and this makes for great problems. So, and then the other thing that came out of our lesion work in with the green anolis lizard was that, just as in the squirrel monkey, we call what corresponds to globus pallidus is the paleostriatum. That's very important for displays. And so here you have animals as unlike as lizards and monkeys, primates, in which the striatal complex is one word I give it to cover all the forebrain structures, or the R complex in a comparative sense, is important for these two basic kinds of prosomatic behavior, what word I use for nonverbal behavior. Routines, subroutines, and then there are four main types of displays, which we all have. And one of them nobody ever thinks of, but there's the territorial, the aggressive display, the courtship display, and what we call the signature display, the John Hancock display. This identifies who you are in the retina. And then there's the other display that particularly psychiatrists, I've discovered, never think of. And that's the display without which none of us could survive, which is the assentive display. Yes, sir. Yes, sir. Yes. Or yes, ma'am.
Yes.
Yeah. So this is, here we have structures that we know are corresponding in reptiles, birds, and mammals because they have the sort of the same histochemistry, and just it extends beyond the cholinesterase and all that business. The dopamine and many other things which are rather peculiar to these structures. So the triune brain model that we've talked about, people refer to it as MacLean's model. I counter by saying it's nature's model, because you can demonstrate it so easily, the corresponding structures as far as the anatomy and the chemistry, the whole shebang, right?
That's right.
And this should be the limbic system.
Yes. And this should be emphasized because most people have felt that this was something that is, it's really a description more than a hypothesis which has to be tested, because the facts are there. It shows that there are these three very different kinds of anatomy which cross the barriers between phylogenetically different species.
And there is this thing, willy-nilly, of directional evolution.
Could you define that a bit more, how you use that term?
Well, I think the therapsids are the easiest illustration, because you had back 250 million years ago when they began to become very prominent and were spread all over the world, when we just had one big continent, which Wegener called Pangea. These fellows were everywhere. But there were many different lines, and they were all going in the same direction. The reduction of the phalangeal formula to the 5 fingers and 5 toes, and the same number of joints and so forth, and the teeth and everything, but most importantly the ear. They were all the middle ear, the stapes and the malleus. I meant to say the incus and the malleus were on their way migrating, but they hadn't gone then to the middle ear. It was still part of the jaw joint. They were still part of the jaw joint. And so Everett Olson said, what do we have here? 2 systems? You know, he was thinking in terms of neo-Darwinism, and then this other business of directional evolution. What do we have here? 2 systems, one for now and one for the future? And as Harley Shands was saying, the wonderful thing about the human is that have developed language which allows them to invent the future. So, you know, but with the limbic system also, that was Broca's great contribution to show that the limbic lobe had been described before, but it was his great contribution to show that these structures are found in all mammals. The rhinencephalon, the great limbic lobe of Broca, forming a frame around. And incidentally, I didn't invent the term limbic system. I just lifted it out of Broca and used it to apply to not only the structure, the cortex of the limbic lobe, but also the structures of the brainstem with which it was connected. Primary connections, you see. Okay. Everybody, as you were saying, they've got the limbic system taking in the whole damn brain, but there are limits. And I think if you sort of define the limits as the primary connections, it helps you out, plus the chemistry, plus the embryology and all these other things, why then it's not so hard to define systems.
No. In defining these systems, you have pointed out a very important fact, and that is that the evolution of certain key behaviors, such as maternal functions, seem to be associated with the evolution of the limbic system in particular, because as far as we can tell, there is a distinct difference in maternal functions in the lizards as opposed to the mammals. What is there any equivalent to the limbic system in submammalian species?
In both the reptile and the bird, you have a very rudimentary cortex, but in the subcortical structures, you can say, well, this is part of the limbic system. But as LeGros, Clark, and Meyer pointed out, there doesn't seem to be anything in the reptilian brain that corresponds to what I call the Thalamus-Cingulate division.
Exactly.
The part of the limbic system which comprises the Cingulate Gyrus. Cingulate means forming a girdle around the corpus callosum. We're talking about that kind of a girdle, and the thalamus. And this sort of appears as a new development in the mammalian brain. But, you know, the things that we discover, it seems, are always so close to our chest we can't see them. And it's struck me as so ridiculous that it took me all my life to discover that the female is the most important thing in the world as far as mammals are concerned. The history of the evolution of the mammals is the history of the evolution of the family. And the female is central to this, because if she leaves her brood, they'll be dead.
Nothing's left.
Yeah, yeah. Whereas the reptile, and they, well, let me just say first of all that the things that I thought I discovered about the difference between reptiles and mammals was lying right there on a plate in front of all of us all the time. Of course, it's the maternal nursing and maternal care, they go together. And very importantly, there's the audio-vocal communication.
Yes.
People say that crocodiles vocalize and so forth, but that's a walking bird. And then the other thing, which nobody, I seldom ask people, that they will come up, and these are people who are professionals in behavior. Play, P-L-A-Y. Those three characterize the evolutionary transition from reptiles to mammals. And this seems to go along with the development of that third Thalamus-Cingulate division. There are three, you know, I've sort of divided the limbic system into three main divisions. And you can see some of this maternal care and so forth cropping up in the septal division and so forth. But the real big development, and it's become enormously developed in the human brain, is the Thalamus-Cingulate division. And this, of course, ties in with the frontal cortex out here. And that's a big place. And there, it seems, we have the capacity to generalize from the family, as it were, to the large family, community family, national family, world family. This brain seems to, this frontal cortex seems to worry about everything.
I'd like to raise a final question in this marvelous discussion we've had. How would you define the mind?
Well, I don't like last questions. they seem too final. But, you know, as you pointed out yourself, what the brain is made of is very soft and gel-like. How do you describe it?
A viscoelastic gel.
Viscoelastic gel. compared to our instruments of precision, which scientists have so much faith in. You know, physicists are always giving hell to the philosophers because they just sit in their armchairs and think about how things ought to be, whereas they have their instruments which will give them precise measurements and so forth. But they seem to forget that the brain always stands between us and what we observe. And so I don't know what in the world is up here, really. Time and space are also purely informational things. Space doesn't exist. Time doesn't exist. They're the transcendental aesthetic of Immanuel Kant. Isn't that true?
That's correct.
And the same with so much else. The brain has got to put it together for us. And in other words, the brain always stands between us and what we observe. And to me, that is a pretty helpless situation, and a very lonely one.
So your definition of the mind basically is saying that whatever the mind is, it's inseparable from this subjective being that we are, that you have to somehow explain how it is that the subjective state, which gives you what Nagel has called the view from oneself, really connects to what scientists try and do, which is the view from nowhere, where everybody can agree on.
Yeah.
And that is really the fundamental problem. How do we fit that together and explain that?
And as I say in this book that's in press now, I refer to this getting to know, the knowledge of subjectivity is the opposite in a sense from epistemology. The epistemology is the outside world looking in, trying to figure out what's going on, but then there's this inside world which is sort of nuclear to all of the rest of the business, and this is the subjective self.
Right.
And this, of course, is what Descartes was saying when the only thing I'm sure of is that I know. But later he added, I feel. But the limbic system is very important, and he didn't know about the limbic system. It's also, there's a precondition. You know, there's this expression, something does not exist until you give it a name.
Right.
But there's a precondition for that. And something doesn't exist until it's touched with a little bit of affect. And that is limbic. I get a little disturbed at times because if you look at the limbic system, you realize that you really can't believe anything. Your intellectual beliefs, you can't believe anything in an intellectual way because, take psychomotor epilepsy. During the fit, the aura, you have this feeling, this is the truth, the absolute truth, and nothing but the truth. And yet this feeling is not attached to any particular item or theory or concept or anything of that sort. Yes. But we know if that thing is blacked out as in a storm through a propagating after discharge through the limbic system, you can go on as a doctor and deliver a baby, you can fly an airplane, or be an engineer and do a very good job getting, I saw such a patient, going from 125th Street to Grand Central. Went through all the lights perfectly, got off, and had no memory for it afterwards. And I've suggested that one of the important things here is that you've got to have a combination of signals from the outside world as well as from the inside world to feel this identity as an individual. And that anterograde amnesia develops with lesions of particularly the frontotemporal structures of the limbic system, because there's no place to deposit a letter, as it were.
That's right.
You've got to have the feeling of an individual, which is very important. But then there's this other very discouraging thing about the brain, and that is that it is like any reasonably complex system. It has to rely on self-reference. So it has no yardstick of its own. So where do you begin with that sort of thing? It's an infinite As Bernowski says, it's an infinite hall of mirrors, this self-reference business.
The problem of infinite regress in mathematics.
Infinite
yes.
regress, infinite regress. Yes. But even, it seems to me, if you multiply the hall of mirrors by 3, it still doesn't add up to as unhappy a situation as this limbic brain, which can't, as far as we know, there's no evidence that it can read or write or communicate under verbal terms. It decides what is real, true, and important, all apart from these emotional feelings that we have. And we shouldn't forget that every feeling that generates, apparently, because it seems to guide behavior on the basis of emotional feelings. Every feeling we have is touched with some degree of affect. Yes. And here we have essentially what amounts to being tried by a jury, having our ideas tried by a jury that can't read or write. It's one thing to have this kind of a brain can identify, yes, that's a bit of food, I can eat it. Yes, this is a person that of the opposite sex, we can get married, something of that sort. But when you're having your ideas tried by this same jury, what can you believe, really?
Yes, I remember you caused a lot of discomfort to the philosophers in town when at the Washington Philosophy Club you presented this paradox, when you said that it seems the only part of the brain from which we get these absolute feelings of certainty is this limbic system. And the limbic system seems to be deciding what is really true and important and beautiful. And since it is our own decision, what independent standard it is, other than perhaps comparison with other standards and making some kind of common ground. Maybe that may be the resolution of this paradox. But they felt very uncomfortable about that because they were much more familiar with the concept of a rational, analytic mind that is superior to the emotions, rather a Platonic type of approach to the problem. So I think you have raised a very important paradox that has to be resolved at the neuroscience level as well as at the level of analytic philosophy.
Here you're looking at a person who really doesn't believe anything. But I do, as a subjective individual, I do have this subjectivity, which is very important to me. And the one thing that I find very reassuring about evolution is whatever else is going on, whatever the hell else is going on, we have up here a new development of which we can't trace any history in the biological world, right?
Right.
With a human being. And I think it goes back much longer, this development up here, than people have talked about. The wonderful thing to me, and mind you, I live in the area of the National Institutes of Health. And to me, this has been one of the most marvelous developments in civilization. There's no history like it. And it's too bad it's become managerial, because we have this wonderful complex of institutions which is not only concerned about other human beings, but also human beings that haven't become sick or old yet. But not only human beings, they're worried about animals too. And it's very hard to live in this day and age and just not have to think about animals all the time if you're in research. Not only animals and human beings are we worried about, but also the botanical world. As you get older, one of the nice things about growing older is you get to be a nicer mammal all the time. But even picking a flower, I'm wondering, well, gosh.
What am I doing? Yes,
What
disturbing the universe.
What am I doing here?